Hi and welcome to Learn Biology with Becky. This is one of our series of walkthrough papers, exam papers that is for GCSE. I hope this really helps you. Best of luck with the revision. Please get in touch. Don't forget to like us and don't forget to subscribe for more of these videos. Thank you.
Okay, so this is going to be a walkthrough of the IGCSE Cambridge multiple choice paper. This is a higher tier paper, also known as extended. You've got 45 minutes to answer 40 multiple choice questions. And this was May, June 2017. Okay, let's start. Okay, question one. Which characteristic do all living organisms show? So this is a Mrs. Gren question. They're very common on these IGCSE multiple choice papers. Obviously, Mrs. Gren is the acronym to remember the 7 characteristics. The E being for excretion, so B is the answer to question 1. Okay, question 2, we have got 4 animal cells seen under a microscope, and the question is, what will be present at X? Now, these are animal cells. So first of all, looking at our 4 answers, 2 of the answers mention cell walls. Now, as these are animal cells, there are no cell walls. So straight away, we can cross through B and D. And don't be afraid to do that on your exam paper. Although you won't answer on the actual question booklet, you will have a sort of a grid where you mark through the letter instead. There's no reason why you shouldn't write or cross out answers to help you when you are actually trying to work out which is the correct letter. Okay, so what will be present at X? Well, we've got 2 cells, so there should be 2 cell membranes. So the answer is C for number 2. Right, number 3, each of the diagrams shows an area of cytoplasm. So we are within a cell. Now, within these cells, there are various what we call organelles or cell structures. This is actually what we call rough endoplasmic reticulum, and the little dots are ribosomes. These are mitochondria, where respiration takes place. Okay, there's 5 of them. Here we've got what looks like a heap of spaghetti noodles, but actually it's supposed to be genetic material, DNA, or it could be RNA, but it's some kind of genetic material. And then in the final area of cytoplasm, these structures are actually chloroplasts. The little stacks, they look almost like stacks of coins, are what you see on the inside of a chloroplast, where the pigment chlorophyll is actually found. So it says, which is from a prokaryote? Now, prokaryotes are basically the bacteria, bacterial cells, and bacteria would not have rough endoplasmic reticulum or mitochondria or chloroplasts. So C, it's a tricky one, but C is the answer. And also they have DNA that's organised just free in the cytoplasm. It's not enclosed in a nucleus. There's no membranes around its structures, whereas in both A, B, and in D, of course, as well, all of these structures have membrane around them, which is shown by the lines. So this is the only one without membrane-bound organelles, so it's a prokaryote. The others are from what we call eukaryotes. Okay, question 4 then. We've got a section through a root, and it says, what are the levels of organisation of the labelled structures? So R is the whole root. P are the tiny little hairs coming off the root. Q is the sort of inner part, known as the cortex, which is this area around the actual vein or the vascular tissue. So first of all, to make life easy, these tables are tricky and people tend to get in a bit of a mess with how to answer these. What I would do is try and identify one of these letters and first of all choose that column. So for R, it's actually inclusive of the whole root. Now the root is an organ. So if we then go to the table and look in the organ column, if we're looking for one that says R, there's actually 2, which is annoying. However, what that does mean is, let's get rid of A, because that can't be right, because there's no R in the organ column, and also the same for D. Now we've got B and C to work, so at least we're down to 2, and we've kind of halved the answers there. So here now, we've got R sorted. So let's look at the other 2 columns for the cell and for the tissue level of organisation. Well, I said to you earlier on that P is the little root hairs. They're actually called root hair cells. Whereas Q, this is going to be made up of lots of cells together. So that's going to be tissue. So we're looking now for P to be under cell and Q to be under tissue. So that makes the answer B for number 4. Right, number 5 then, it says which characteristics are correct for both osmosis and diffusion? Again, it's one of these table questions that people tend to find quite tricky. Let's find something you know, all right? You should know, hopefully, that osmosis and diffusion are both what we call passive. That means they don't require energy, okay? So if we go to this column, are they energy consuming processes? No, so we're looking for the cross. If it's got a tick, let's eliminate it, get rid of it, no good to us, can't be the right answer because already in one of the columns there's an incorrect answer. All right, now that leaves us with A and C. So we sorted this column, let's give it a tick. Let's go to the next column, require a concentration gradient. Right, okay, so unfortunately the answer's the same in both, so that's no help to us. Let's go to the first column, require a partially permeable membrane. Well, the answer is either they both do or one or both don't. Now, actually, diffusion does not require a partially permeable membrane. If you spray some nice kind of scent, your perfume, your body spray, whatever, it will diffuse across the room, regardless if there's a partially permeable membrane present or not. Osmosis does need a partially permeable membrane, but they both don't need it. Therefore, we can go for C, and that is our answer for question 5. Brill. Okay, so number 6, which diagram shows the structure of DNA? Straight away, again, hopefully, C and D might look a little bit odd to you. These are the bases, all right, represented by the letters A, T, C, and G. They're kind of on the outside of the DNA, which doesn't look quite right. So let's exclude those and let's focus on A and B. Now, what you need to be looking at is the pairing of the bases or the letters, as you may refer to them as. A and T always go together, so it's remembered as apples in trees, and C and G always go together, so you can remember cars in garages. Any little kind of memory aid is great for obviously trying to remember all of this factual stuff in the GCSE course. So if we look at this A, the first picture, A going with C can't be right. A will not go with C, it will only go with T, or T with A, so that's got to be wrong. If we look at B, they all look good. A with T, C with G, G with C, so yeah, that one's good. We're going to go with B for number 6. Okay, number 7 is about enzyme activity and how it's affected by temperature. So we've got a little graph here. Temperature, as it increases, what's happening to rate of reaction? So to begin with, obviously temperature's going up. And then at around about 40 degrees, we're seeing a massive drop in the rate of reaction. And the question is, how can the change in activity between 40, so here on the graph, and 55, around about there on the graph, be explained? So this big drop in the rate of reaction to the point where the enzyme is actually not doing anything anymore. What's happening? Some of this is about wording, so just be careful. The first answer, heat has killed the enzyme. Definitely not. You cannot kill an enzyme. It's not a living thing. It's a chemical. It's like saying you could kill, washing up liquid. It's nonsense. So just watch that. Enzymes do not get used up. Right? They keep getting freed up, and they can keep carrying on catalyzing the reaction. So that's a definite no-no. Reactants are moving faster. Now, if the reactants were moving faster, we'd actually probably see the rate of reaction actually increasing, going upwards on the graph, as it does to begin with. So that's not the right answer. So the last solution is correct. The substrate is less likely to fit into the active site. That's basically describing, if you like, the what happens through denaturation. So denaturation, or when the enzyme denatures, means that the substrate will no longer fit into the enzyme's active site due to a change in shape. So D is the correct answer for number 7. Right, number 8 is a really tricky question, and people really stumble with what's going on here. So let's just go through what we're being asked first of all. We've got 4 tubes set up. It says which one will contain the most carbon dioxide after an hour. Now, A and B have both got some kind of black, like bin liner material around them. Polythene, it's just like plastic bag sort of stuff, whereas C and D don't, and light can get in. So we do a little talk through what's happening. In A, you've got both a snail and a bit of pondweed, an aquatic plant. And both of these organisms will be respiring. Plants respire all the time, as do animals, to get the energy they need to live, to do what they need to do to keep alive, and so on. However, they're both making CO2, but due to the fact that light cannot get in, it's being stopped by the black polythene, the plant will not be photosynthesizing. So it won't be actually taking any of that CO2 out of the water for photosynthesis. That means 2 organisms producing CO2, but none of it being removed, it's going to be pretty high in A. If we move on to B, same setup, but we're lacking the snail. So again, we're going to have carbon dioxide being produced by the plant, but it's not being absorbed by the plant for photosynthesis because no light can get in. However, there's no snail. So in theory, B won't have as much carbon dioxide as A. For C, just a plant on its own, it will be respiring, but light can get in, so it can photosynthesize. That means CO2 being made, some of it will be used by the plant, so it should be fairly low in CO2. And finally, we've just got a little snail on his own. He'll be respiring and making CO2, and therefore there will be some CO2 production, but not as much as in A, where there are two organisms both respiring and no photosynthesis. Really tricky question, that one, and it really gets people a little bit confused. So well done if you got it right. Okay, number 9, another tricky question, a data question. We're using, we've got two sets of data on the same graph, which is always a little bit confusing to look at. So we have daily CO2 daily CO2 uptake, which is the darker points here. Okay, there's your CO2 uptake. So this is from sort of midnight through to 12 o'clock noon through to midnight the following night. And then we've got transpiration, which is the clear or white dots shown. It says, what can be concluded from this graph? That more stomata are closed during dark periods. Now, during dark periods on your graph, so this is like from midnight to early morning, this is dark, okay? This section here, let's just draw in so you can see, all right? This is when it's going to be dark. And similarly, when you get to this time, 8 o'clock at night onwards till 12 o'clock, midnight, that's also going to be when it's dark. So when it says more stomata are closed, let's see what's going on. We've got quite high water loss. And we've got quite a lot of CO2 being taken up because both these lines are highest on the graph at the times when we would think it would be dark. Now that would suggest they're not closed because water can get out and CO2 can get in. Okay, so A doesn't look quite right to me. More stomata are closed during the light periods. Now, the light period is this section of the graph that I haven't put a yellow line next to. So whatever time, 6 o'clock in the morning, depending on the time of year, through till 4, 5, 6 o'clock in the evening or later in the summer, we've got very low rates of transpiration. That means the plant's not losing a lot of water through its stomata. We've got very little CO2 uptake. That would suggest to me the stomata are closed at this time, and therefore I would circle B. Now let's just check C and D that there isn't anything there that might make us change our mind. So C, there is no carbon dioxide uptake during dark periods. Well, at no point does the line for CO uptake touch 0. So that's not true. That cannot be the case. There's always some CO2 uptake. It just fluctuates during the day, night, etc. And finally, there's no water uptake during light periods. Well, there's always transpiration, which isn't actually water uptake, that's water loss. So we can't actually know that from the graph, so can't be concluded. Therefore, I'm happy to say the answer is B. Okay, let's go to number 10. It's a bit more straightforward this time. You either know it or you don't, or you're going to have a guess. Which ions are used to make amino acids? Now, amino acids are the building blocks for proteins, and hopefully you remember that proteins are a little bit different to carbohydrates and lipids because they contain the element nitrogen. And nitrogen gets into the plant in the form of nitrates. So the answer to 10 is B. Right, 11. We are a quarter of the way there. Okay, following symptoms can be caused by a dietary deficiency, so a lack of something in the diet. If we look through all 4 bullet points, it's something related to either our bones or our teeth. Again, 3 of them are down to bones. 1 of them is to do with the teeth. And if we look through iron is all to do with red blood cells and linked to anemia, so not likely. Protein, that's more to do with kwashiorkor and, lack of growth and so on, so unlikely. Vitamin C, linked to healthy connective tissue. Deficiency diseases, scurvy, unlikely. And that leaves us with vitamin D. Vitamin D and calcium, which is a mineral ion, work together. Both of them are needed really for absorption to enable us to have strong bones and teeth. All right, you might have seen that on adverts for dairy products. Okay, moving on to question 12. Ooh, cholera. Right. This is quite a tricky little part of the human nutrition topic. So we just need to get them in the right sequence. The answers are there. We're just getting them in the right order. So if we go through, first of all, chloride ions are secreted into the gut. Osmosis causes water to move into the gut. The infected person becomes dehydrated. Toxins are produced by the pathogenic bacteria. Okay, so we're going to put them in the right order. First of all bacteria produce toxins. That's how they make us ill. So this is going to be our starting point. So this is going to be number 1. It says then we've got to choose one of these as our number 2. So I would go with chloride ions are secreted into the guts. What they do is, that's not a very good 2, is it? What they do then is they dissolve in the contents of the gut. They lower the water potential, which then draws water by osmosis into the guts. I'm not sure if I'm going to be able to write a number 3. Yay, okay, there's a number 3. So we've got 1, 2, 3. And then finally, due to the fact that that person's losing water from the bloodstream and their cells into their intestine, which will be passed out via their sort of feces, their diarrhea, this is going to cause dehydration as the final step. So yeah, can't draw a number 4. So let's have a look. 4 should be first. So let's eliminate A and B. All right, then it should be, oh no, my letters look the same. Sorry, yeah, number 2. So it should be second one should be number 1. Oh, same order. Okay, so we haven't eliminated one of these yet. Then the next thing is it should be number 2. Osmosis causes water to move into the gut. And finally, 3 should be at the end. The infected person becomes hydrated. All right, hopefully I didn't confuse you too much there. Sorry if I did. Right, moving on, 13. Right. Human tooth, and it's 4 types of teeth that you should know about. There are the incisors at the front, then the canines, then the premolars, then the molars. Now, because this has more than one root, we can assume it's one at the back of the mouth, either a premolar or a molar, and their job is to crush or grind the food, increase its surface area. The teeth at the front, like the incisors, are more the biting and cutting teeth, and then the canine is the one we use for tearing meat off a bone, or I don't know, crusty baguette, whatever it might be that you are eating. Right, 14. Enzyme question. Okay, so the graph shows the effect of pH on the activity of 3 different enzymes. Not only have we got a graph to look at, we've also got a table just to make things a little bit tricky for us. So first of all, let's match up what's going on between the graph and the data in the table. So part of the alimentary canal is the mouth, 7 pH. Okay, so this is going to be letter Y. This is going to be the one that's working in the mouth. The next one in the table is the stomach, 2, so pH 2, that's X working in the stomach, and then small intestine, 8.5 up here, so Z is this one that's working in the small intestine. Then the question is actually asking which enzymes in the graph are likely to be protease enzymes. So you need to know your protease enzymes, of which there are 2. The first one is pepsin, and pepsin is very special because it works in the stomach at a very low pH. So that's pepsin, that's a protease. Your other protease works in the small intestine, and that's called trypsin. Your pancreas actually makes it, but then it works in the small intestine. So we're looking for the one at 8.5. So that's X and Z. The other one, Y, is working in the mouth. Well, there is no proteases made or secreted into the mouth, so that's a no-no. So we're looking for X and Z. Let's go along. All these letters are looking very similar, so just be really careful making your selection. On these kind of papers, just a few marks can make the difference very easily between one grade and another on the multiple choice paper, because it's only out of 40 marks. Okay, we're on to question 15. Right, we've got a section through a root, so we're back to plants again, and the section through the stem, and we're just looking for the arrangement of xylem and phloem, really. Xylem, I always remember, is it marks an X in the root, so the X marks the xylem, or X marks the spot right in the centre, so W is definitely xylem. So if we look at our table, we need the right combination across all 4 columns. So W has got to be xylem, which means I can eliminate C with a very wonky line. I can eliminate D with another very wonky line. Luckily, it straightens itself for me. Now, I am left with basically this diagram to help me decide between A and B. So these 2 are the same, all right, so that's no use. So which way round? Now, in the stem, the xylem is always towards the middle. Okay, like it was in the root, it's more central into the centre of the stem, and the phloem is on the outside. I remember it as P for periphery, meaning on the outer part. P for phloem, P, easy for me to say, P for periphery, whatever works for you. All right, so we are looking for Z, xylem, and Y as phloem. So let's have a look. Z, xylem, Y, phloem. Take your time because everything's all a bit confusing. All the letters are very similar and you've got all the same words just jumbled up. It can really throw people, so really take your time. So we're going for B for question 15. Right, you do well, guys. Well done. Okay, number 16. Okay, what processes are used by root hairs to take up ions and water? So two separate things were being asked here. First of all, how are ions taken up? And secondly, how is water taken up? There's only one way water moves, and you've got to learn this, and that is by osmosis. So we've got two answers in the table that say osmosis, which are correct. Let us eliminate B and C. Bye-bye, B and C. With ions then, they are actually taken up by active transport. If you look at D, it's got osmosis for both. Now, osmosis is purely about the water, about the movement of water. So it has to be therefore A. Ions are taken up by active transport from the soil, lowers the water potential in the root hair cell, and water will follow by osmosis. Right, that's A for 16. Okay, moving on to 17. We're nearly halfway there, not quite. Which of the following increases transpiration? Now, transpiration is water loss from the leaf through the stomata. So we're looking to find one of these that increases the water loss. So let's have a look through air around the leaf with high humidity. Now, that's actually going to decrease transpiration because if it's quite humid around the leaf, there's not going to be much of a gradient, if you like, a difference between the inside and the outside. They're going to be fairly similar. So that's going to decrease transpiration. So that's a no-no. The molecules around the leaf have less kinetic energy. That's kind of a very posh scientific way of just saying it's not very windy. Now, actually, wind or air movement increases transpiration. So if it's not moving around the air much, it's not very windy, it's still, another way of putting it, it'll actually decrease transpiration. So that's a no-no. An absence of light falling on the leaf. Now, when there isn't light on the leaf, the stomata tend to close to try and prevent too much water loss. Therefore, that would decrease transpiration because if the stomata are closed, the water can't get out. And finally, well, we've kind of done it already. We've eliminated the other three. So D is the answer. Water molecules in the leaf with more kinetic energy. So the actual water in the leaf, if that's got more kinetic energy, they're more likely to move out to the surrounding air, and that will increase transpiration. Okay, a human blood cell, what is its function? Now, if you can identify it, that might help. So this is actually what we call a phagocyte, and you can recognize them because their nucleus, this part here, is what we call multi-lobed. So it looks a bit like three kind of sections, just with these very sort of slight connections that you can see. Now, the reason for that is they can actually, they're very flexible. They can squeeze through the capillary walls and get through to tissues where there might be infections. So it's a phagocyte. So if we go through the functions, antibody production, no, that's the lymphocyte's job. Fibrinogen production is all about your platelets in your blood. Oxygen transport, red blood cells, that's definitely a no. So the final one, I told you it was a phagocyte. Phagocytes carry out phagocytosis, where they engulf pathogens into the cell and destroy them. So let's go with D for 18. A disease-causing organism is called a pathogen, all right? Antibody is what we make against pathogens. The host is us. If we're infected, we're the host. We're hosting, not a nice party, but unfortunately we're hosting the pathogen. And a phagocyte, I've just mentioned in the previous question, is a type of white blood cell that carries out phagocytosis. So C is our answer. Okay, 20, we're halfway there. Well done, guys. Which is a function of the cartilage in the trachea. Is it for gas exchange? Well, that's the job of the alveoli, so that's a no. Does it prevent collapse of the trachea? That sounds good to me. I'm going to circle it for now. I'll come back, just check there isn't anything else. Produces mucus. No, that's for the goblet cells. Does it trap bacteria? No, that's what the mucus does. So B, I am confident with that answer. Okay, 21. We are over the halfway mark now, so brilliant. The equation for anaerobic respiration in yeast just looks like a big jumble of letters and numbers. Happy days. Let's work our way through. Well, straight away, actually, I recognised it, but I'm just going to go through anyway. Anaerobic doesn't require oxygen. So there's one that says oxygen is included as a reactant on the left-hand side. That's a no-no. It's anaerobic, so it's without oxygen. So let's get rid of B. And then that's okay, there's no oxygen. This one, we're saying we're using CO2, which seems a bit nonsense. And you're using CO2, and then you're actually making glucose. Nonsense, let's get rid of that one. This one is also using carbon dioxide plus water to give you glucose plus oxygen. Now, you might even recognize this if you've been revising hard. That's actually the equation for photosynthesis. So that can definitely not be the answer. So A is showing us that glucose is converted to ethanol and carbon dioxide. The numbers in front are just to balance the equation. So you've got the same numbers of carbon, hydrogen, oxygen atoms on the left-hand side as you do the right-hand side. Okay, 22. We've got a section through the kidney from top to bottom, and there's some labelling. Now, again, looks all very similar, so we're going to pick this apart, take our time. Okay, first of all, let's have a look at A. Renal artery going towards the kidney. So that's correct because it's coming from the heart, away from the heart, A for artery, A for away, to the kidney. The ureter is coming away from the kidney and carrying the urine. All looks good. Ureter is right. Cortex on the outside of the kidney, medulla, meaning sort of middle or central. That actually looks really good to me. I'm going to circle it because I think that's the right answer, but I'm happy just to go through. Let's have a look. Okay, B. The error that's glaringly wrong to me is the fact that it's got the renal vein bringing blood to the organ, to the kidney, when actually the vein would be taking it away. So no, not happy with that. The other bits are all okay. It's just this bit that makes it wrong. And then C, we've got these the wrong way around. The cortex is always the outer region, and same with D. So yeah, A is the answer. Okay, question 23. Reflex actions. It's one of these, we've got to put them in the right sequence. Only 3 points to get in order this time, so maybe a little bit easier than the cholera question. Let's have a look then. Nerve impulses pass from the sensory neuron to the relay neuron. Nerve impulses pass from the motor neuron to the effector. Nerve impulses pass from the receptor to the neuron. So it's about the starting point. The starting point is always a receptor, which is in the sense organ, whether that be the skin, eye, whatever it is, okay? So that's going to be our first one. So number 3 even. So we're looking for one with number 3 at the beginning, all right? That's either C or D. That means these can't be right, so let's get rid of A and B. And then we're just picking between these two, which one comes next. Now, the effector always comes at the ends. And the sensory neuron would come from the receptor, pass it to a relay neuron in the CNS, and then pass it to the motor neuron and then to the effector. So we need it to go 3, 1, then 2, 3, 1, then 2. That's it. C's our answer. Real. Okay, 24. Still on the nervous system, but this time on the eye specifically. We're looking for how many different kinds of cone, how many different kinds of rods. There's only one type of rod receptor, so let's get rid of B. Let's put a line through it. Let's put a line through D. Okay. Now, we've got to decide, is there one type of cone or are there 3 types of cone? Well, we have what's known as trichromatic vision, meaning 3 colours, and that's down to the fact we've got 3 types of cone, which are for the 3 different colours. So C is correct. And then 25, what happens when we release adrenaline? There's 2 factors we're going to look at. What happens to blood glucose? What happens to pulse rate? Now, this one's probably the easiest one to start with. If you think of adrenaline being released, you're scared, you're worried, whatever it might be, someone's just made you jump, your pulse rate's going to suddenly increase because your heart rate's going to increase. So we're looking at B or D. So let's just get rid of, crossing them out really helps. It just really focuses you then on the 2 that are left. It really, this kind of eliminating 2, it's a bit like Who Wants to Be a Millionaire? It's gonna really help you in your technique of answering these papers. So we've got, is the blood glucose concentration going to go down or is it going to go up? Well, adrenaline is all about preparing you for activity. You want your blood glucose concentration to go up. Then there's more glucose available for cells, particularly muscle cells, for respiration. So it's increased for both. The answer is D. 26, which statement about microorganisms is correct? Bacteria are killed by hydrochloric acid in the duodenum. Okay, it was all right until we got to duodenum, because no, we actually have it in our stomach, whereas the duodenum is the first part of the small intestine. So nope. Bacteria can become resistant to antibiotics. Sounds good to me. I'm going to circle it for now. Bacterial infection and viruses are treated with antibiotics. Definitely not. These, probably know all this anyway, if you ever go to your doctor when you've got the cold, when you've got a cold virus. Absolutely not. You cannot use antibiotics against viruses, only bacteria. And finally, bacteria produce sugars that cause tooth decay. Ooh, that often gets people. It's because they actually break down sugars, but they don't produce. They break down sugars and they produce acids that cause tooth decay. So that is also wrong. So B is the answer for 26. Okay, genetics question. So we've got flowers that can be 3 different colours, red, pink or white. A red flower is always homozygous, which means it's got 2 alleles the same. Same for the white flower. Pink flowers are heterozygous. This question is about codominance. So when you cross a red and a white flowered plant, you will get the in-between, the intermediate, the pink. And therefore, if you cross a red and a white flower, what offspring will be pink? What percentage of offspring will be pink? It will actually be all of them. Each offspring of the red and white parent plants will have 1 red allele, 1 white allele, and therefore will be pink. Okay, an alligator has 32 chromosomes in each of the cells of its nose. How many chromosomes will an alligator zygote contain? It's a bit like a riddle. Now, 32, and a zygote is actually diploid, so it has both sets of chromosomes, so it will also have 32. Don't be tricked by CIE. Don't let them play tricks on you, okay? Okay, question 29. Which hormone is produced by cells in the placenta of a developing fetus? Well, FSH and LH are both hormones produced by your pituitary gland, so to those. Testosterone, no. And progesterone, therefore, is the correct answer. The placenta produces progesterone, maintains the lining so the pregnancy is not lost. Okay, this should be quite a nice question on contraception, which is not a barrier method. Right, A, male condom, definitely. So we're looking for the one that's not, by the way, so just read it carefully. B, this is the femidom, so this is also a barrier method. C, doesn't really look like any of the others, so let's come back to that in a minute. And D, this is actually the diaphragm or cap, also a type of barrier method. This is the one that sits inside the uterus to prevent pregnancy, an IUD. So this one is not classed as a barrier method. It does not stop the male seminal fluid from obviously entering the woman's body. Okay, question 31. In someone who is HIV positive, which part of their blood is destroyed? It's where the lymphocytes are actually reduced to very low numbers. So that is the answer for 31. Okay, what are stem cells for 32? We are into the final 10 questions now. Are they specialized cells or are they unspecialized cells? Let's just look at this first part of the sentence. Well, hopefully you know stem cells are really, really important because they haven't yet specialized. They have the ability to become, some of them anyway, nearly any type of cell in the body. So A and B, let's get rid of them, all right? They can't be specialized. That means they've already become a particular type of cell. Therefore, they don't have the potential to become different types of cell. Not much good for us then. So they're unspecialized. Do they divide by mitosis to produce daughter cells, or do they divide by mitosis? So we're looking between these two words. Meiosis is all about producing gametes. So that's your sex cells, your sperm, your egg cells, and so on. So no, that can't be right. Not meiosis. They divide by mitosis, just normal cell division. Okay, 33. The list shows the diploid number of chromosomes of 4 organisms. There they are. Fruit fly 8, human 46. You should know that one. Potato 48. Shame that we don't look that similar, considering we've got quite similar chromosome numbers. And the garden pea is 14. What is the correct chromosome number of a male sex cell in each of these gametes? So the male sex cell is haploid, not diploid. HA at the beginning of haploid, I remember as half. So all we need to do is halve these numbers to get the answer. So fruit fly has to have 4. So that's nonsense. That's nonsense. Okay. And then if we go through, human has to be 23. So we've already got our answer A. Just if you want to check, 24, half of 48, 7, half of 14. Bam, we've done it. 33 is A. Okay, which of these is a population? All the insects. So a population is all the organisms of the same species in a given area at a given time. So all of the insects, there's going to be lots of different species of insects. I doubt it. Tigers, yes, they are the same species. However, there are subspecies, which is slightly confusing, but they are the same species. And if we've got a specifically named group of trees, we can assume that's a species as well. So I would go for 2 and 3 only. And the answer is D. Last 5 questions, or last 6 anyway. The number of bacteria resistant to antibiotics is increasing. What has contributed to this? So what has actually caused this to happen, if you like? Choosing the antibiotic carefully to match the strain of bacterium. No, that would actually help prevent antibiotic resistance. So let's say no to that. Not completing the course of prescribed antibiotics. Going to put a circle around that one for now, because yes, that is not helpful at all, and that has been known to increase bacteria becoming resistant because it doesn't fully kill them off and so on. Tracing, testing and treating people who have been in contact with the infected person. Well, that sounds like it would be a good thing rather than something that might contribute to something bad like bacteria becoming resistant to antibiotics. Using antibiotics only when essential. Again, that's a good thing. That's hopefully not going to exacerbate that antibiotic resistant. So I'm going to go with B for 35. Okay, now we're into the last 5 questions. Right, of the processes listed 1 to 4 that affect the concentration of CO2, which would increase the concentration? So which would increase? Increased deforestation, cutting down more trees. Yeah, I would say so. All right, less CO2 absorbed by photosynthesis. Increased forestation, the opposite, planting more trees. No. Decreased fossil fuel combustion. Well, if we're burning less fossil fuels, there's less CO2 produced. So no, that's not going to increase the concentration of CO2. If we, however, burn more fossil fuels, yeah, that's going to increase CO2. So 1 and 4 gives us the answer of B. Okay, last little section now. Graph shows the changes in the population of predator and prey over a period of time. Which point on the graph shows a decrease in predator population? First of all, you need to identify which of these lines represents the prey and which one represents the predator. Easy way to remember it is there's always less of the predator. There'll be more of the prey. Just in your general food chains, because there's so much energy lost as you move along a food chain, there's fewer of the predator at the next trophic level than the organism below the prey. Okay, so let's look at this curve. That means we can choose now between B and D. Can't be A and C because that's the wrong line. That's our prey, and we're looking at predator. The only one that shows a decrease is B. All right, that's number of them coming down, whereas D is showing their numbers going up. So the answer is B. A reason for using bacteria in biotechnology. They're found inside the human body. They may very well be, but that isn't really a reason for using them. So it seems unlikely. Got to get rid of that one for a minute. Bacteria can be resistant to antibiotics. Not sure if that's really that useful for biotechnology. So let's just put a line for it for now. Bacteria can make complex molecules. Okay. Or bacteria reproduce slowly. Well, actually, the fact they reproduce so quickly, let's cross through it then, is the fact they reproduce so quickly is actually a really useful thing because we can make the products we're getting them to make for us really quickly. Now, this is actually the answer because we can put human DNA into bacteria and they can make complex molecules like insulin, for example, which is a human hormone used for regulating blood sugar in diabetic people. Why is yeast used in bread making? Out of all of these, it's to make CO2. It gets trapped in the dough and then we get that nice spongy bread. It's not too dense. So yeah, that's B for 39. Lots of B's. 40, last question. It always seems to be sewage or to do with sewage, the last question on these multiple choice papers. It says the graph shows the levels of dissolved oxygen. All right, this bit here. And also the mineral ions down here. Fortunately, they haven't used a sort of different dash line or whatever, but just look carefully. All right, it says at which point does raw sewage enter the river? Now, when raw sewage enters the river, generally the oxygen levels go down because of the bacteria breaking the sewage down. They're respiring, they're multiplying really quickly, and they use up the oxygen in the water. What you're going to expect if sewage is entering the water is a sudden surge in mineral ions from the actual sewage. And so I would say with quite a surety that A would be our answer. And that is where the sewage was introduced to the river. And over time, you might see that it recovers back to what it was before the sewage entered the river. Okay.
I really hope that's helped, guys, with your exam technique for the multiple choice paper and also the revision of your biology in the areas that were covered. I've included the mark scheme and also the grade boundaries for that paper so you can get a feel for what you would need to achieve to hit those different grades. Well done, guys, and keep up the good work.